Hello everyone. Today we are going to start the second part of our course talking about specific topics in immunology. So our topic for today is tolerance and mechanism of autoimmunity. And let's talk about tolerance. Tolerance is a state of control and responsiveness. So the immune system recognizes this antigen, can see this antigen, and the immune system decided deliberately not to react to this antigen. So this is the sort of tolerance. So in tolerance, there is no immune reaction, no aggressive response against this antigen. We need tolerance. We need tolerance to our self-antigens. So the immune system is going to recognize our self-antigen and should tolerate the presence of these antigens as being self. And also the immune system needs to recognize or is going to be exposed to certain non-self antigens that it should tolerate, like the normal flora, for example. So it would see the normal flora, it would recognize the presence of normal flora, but it's not going to react to it or interact with these normal flora. So we need tolerance, and this tolerance is controlled and managed both centrally and peripherally. Centrally, we mean the control of tolerance in the primary lymphoid organs like the thymus and the bone marrow, and the peripheral tolerance is the control of immune reaction after the cells, the lymphoid cells, whether T or B cells, exit the primary lymphoid organ, when they are in the circulation, when they are in the tissues. So there should be sort of peripheral control to maintain the state of tolerance. So we're going to discuss these types, different types of tolerance or stages for controlling tolerance. The first and the most important one is the central tolerance. So we've discussed before in the last lecture when we talked about the negative and positive selection for the T cells in the thymus. So the T cells are positively selected, that they have a moderate interaction with the self-MHC. So those cells are positively selected, and the cells that interact with self-antigens, when the self-antigens are presented to them in the medullary cortex. I think I have image for that. Yes, let's see this. So we've saw this before, and we stated that here is the area of positive selection where T cells are exposed to self-MHC, and then in the medulla, the cells would present, there's the medullary thymic epithelial cells, and these cells would present different source of self-antigens. And T cells that are able to interact with self-antigens are not selected, and they would be deleted. So here is the process of negative selection, which is the core of central tolerance. Okay, so we also stated that almost 95% of T cells would die with apoptosis. Only 5% would make it to the periphery here that can exit out after being after passing both the positive and negative selection. So this is the major part of central control of tolerance. The same process occurs in the bone marrow for the B cells to be selected, only B cells that recognize foreign antigen, and those that recognize self-antigens are deleted before going out to the circulation or to the lymph nodes. They would be deleted. So this is the control of tolerance centrally in the main or in the primary lymphoid tissue. So what goes wrong? The first problem that might happen is that not all self-antigens are presented in the bone marrow or in the thymus. And this is a possibility. So the medullary thymic epithelial cells might not be able to present all types of antigens. There are certain types of proteins that are not expressed in the body until the body reaches to the puberty stage, for example, like certain hormones. It might, the T cell that recognizes self-antigen might not come in contact with the medullary thymic epithelial cells. So all of these are possibilities that might happen, and they actually do happen in the normal physiological conditions. So some of the thymic cells, some of the T cells that can recognize self-antigen can escape and go into the circulation even after negative selection. So this is one of the, let's say, problems or the breakdowns where central tolerance goes wrong. Other problem is that there are very few cells are not killed, but they are made energetic. So what do we mean by energy? Energy is a state of immune unresponsiveness. So sometimes in the thymus, when the T cell that has a T cell receptor recognizing self-antigen, usually it should be deleted in the negative selection. Sometimes it would not be deleted. It would become sort of lazy, unresponsive sort of T cells. And these T cells, although they are unresponsive, they are harmless. So even if they face the self-antigen, they would not react to it because they are in energetic state. It is possible that under certain conditions they might become activated, like for example when they are overstimulated by exaggerated inflammatory reaction, or when they are presented at an area where there's huge amount of cytokines, they might become activated. So this state, the energetic state, is one of the possibilities to leave a threat of self-reacting T cells. How does energy occur? So remember, last time we said to activate the T cell we need two signals. We need the binding of the T cell receptor with the MHC, so this is the first signal. And to complete the activation, we need the second signal when the CD28 on the T cell binds with the B7 on the antigen presenting cell. What happens in the case of energy, this T cell does not receive the second signal. So although this T cell can recognize a self-antigen and can bind to antigen presenting cell holding a self-antigen, or maybe binding to a self cell holding a self-antigen in the MHC class 1. So it will bind to it, but it lacks the second signal. Maybe the CD28 is not fully activated. It cannot interact for any reason to the B7 on the antigen presenting cell. Maybe there is other cascades here in the cell that are not completely structured. So still it is in the energy state. This sort of a latent T cell that is not able to react to self-antigen. But as I said, if it happened that it become present in certain situations, it might manage to receive the second signal and got activated. Let's talk about peripheral tolerance. In peripheral tolerance forms an additional mechanism, an additional control to self-reactivity. So if there, for any reason, there is a failure in the central tolerance. Peripheral tolerance would manage to restore or manage these pitfalls in the central tolerance. The peripheral tolerance occurs in the periphery, so outside of the primary lymphoid organ, and there are different mechanisms that the peripheral tolerance follows: the energy, ignorance, and suppression. Energy that we talked about is, again, there is a lack of the secondary signal in the periphery, so the energetic cell that goes out from the thymus, for example, it will remain energetic in the periphery. There would be no stimulation for this T cell to receive the second signal and become activated. Another form of energy is making a B cell energetic, which means, again, the B cell, remember, it requires the T helper cells to complete its activation. It needs an interaction with the helper T cell to produce cytokines to activate the B cell. So sometimes this B cell that holds B cell receptor or antibody on its surface, it can recognize a self protein, but although it can bind with that self protein, it did not receive the signal from a helper T cell. It did not receive the appropriate cytokine from a helper T cell to become fully activated and to react to this antigen. So it is in a sort of energetic state. The second mechanism our body follows to control or to maintain a state of tolerance in the periphery is ignorance. And in ignorance, it means that the amount of exposed self-antigen, although the self-antigen is there, but it is present in a very small amount, that it is not adequate to mount a huge immune response. So it is there. The T cells or the B cells that recognize the self-antigen is also present at the scene, but the amount of that self-antigen presented is actually not adequate. So the T cell, although it sees that antigen, although it can bind to it, but it does not mount any immune reaction and decided to ignore the presence of that antigen. And sometimes these self-antigens that are presented, and the T cells that have the T cell receptor specific for that cell antigen, they do not meet. They simply, maybe because this self-antigen is presented in a hidden area in the body, so they are not present like in the scene when the T cell is circulating in the bloodstream. They would not manage to see that antigen because it is in hidden places, like for example in the brain. So the brain, not all, or there's a blood-brain barrier that prevents the entry of immune cells and reduced chances of the meeting between that T cell receptor, self-reacting T cell receptor, with the self-antigen. And the third mechanism is suppression. What we mean by suppression is to suppress any immune reaction by presence of a T cells that control the immune reaction, which is the Treg. Remember, we said there's one of the classes of the CD4 T cells is the Treg that regulates the immune reaction. There is different mechanism. The regulatory T cells follow to control the reaction, either by direct contact, as in the image we've seen here. So there is direct cell-to-cell contact, and in this case, it will inhibit the activation of these T cells. It would not allow it to produce cytokines or not allow it to proliferate. Sometimes it acts by producing interleukin-10 or by producing transforming growth factor beta. All of these mechanisms are followed to control the immune reaction and make sure that immune reaction would take place only when it is needed, and would not occur just whenever a T cell receptor meet with an antigen, whether self or non-self antigen. It should meet the antigen at adequate amount that forms a threat to the body. At that moment, the immune reaction is allowed to build up. So we've seen how important it is to maintain tolerance, and tolerance is controlled centrally in the thymus for the T cells and in the bone marrow for the B cells. And usually the cells go into deletion, like 95% of the T cells, or maybe 99% of the T cells would be deleted if because they are reacting to self-antigen, or they might go into anergic state. Also in the periphery, some T cells might be deleted or goes into energy state, or might be regulated by T regulatory cells, or might not be able to mount an inflammatory reaction by ignorance, either because the amount of antigen is so small or because it cannot see the antigen. So these are the mechanisms our body do to maintain a state of tolerance. In autoimmune disease, there is loss of the tolerance, and there's something wrong goes here. Usually the deletion will end the problem, but having anergic cells, having hidden antigens, having regulatory cells that are responsible for regulating the immune response. So if there is something goes wrong with these regulatory cells, or the anergic state has changed for any reason, or the self-antigen become apparent to the T cell, then there is a potential for an autoimmune reaction. So loss of self-tolerance goes into 3 stages. The first stage is autoimmune potential, where there is autoreactive T cells circulating in the body, but they are not activated. There is no autoimmune reaction takes place. We are actually all in this stage. We all have the autoimmune potential. The central tolerance mechanisms and the peripheral tolerance mechanisms are not 100% effective. There must be a T cell, couple of T cells, many T cells with autoreactive T cell receptors are present in our circulations. But this doesn't mean they are going to interact or react to these self-antigens. So this is the first stage. The second stage, the physiological autoimmunity, and we call it physiological because it is actually important for for maintaining the effective action of the immune response. In this stage, there is self-reacting T cells, and they actually come in contact with the self-antigen and sort of interact with these self-antigens. However, they did not mount a huge immune reaction. And the importance of this stage is the maintenance of a healthy immune system. So, how does this work? Presume that you are living in a sort of a very hygienic atmosphere. Your body never met with a bacteria, with a fungus, with a virus. You do not have stress that is gonna irritate your immune system, which is almost impossible. And your immune system is just living in peace. Actually, there is nothing to activate or irritate your immune system. What happens is the T cells, the adaptive and the innate immune cells are not acting, are not functioning, so they will go into sort of a dormant and lazy stage. So when your body become faced with a pathogen, it requires time to interact with that pathogen. You need to heat up these cells to go back to work. So this is not a healthy state of the immune system. So to maintain a healthy immune system, your cells, either in the adaptive or in the innate immunity, should maintain a certain level of work, certain level of action, so they can, when needed, they can interact quickly. So this is the importance of autoimmunity. Like sometimes our self-antigens, when they interact with self-reactive T cells, they would create sort of training session to the cells, to the antigen-presenting cells, to the macrophages, cytokines, activating the complement system. So they make things running so that when you are met or faced with a real pathogen, they can come into action quickly. So this is the physiological autoimmunity. It is also important and essential to maintain good healing, better healing for the tissues, as the cells are always ready to conduct their action and recruiting inflammatory cells for healing process. So this is the physiological autoimmunity, which again most of us are using to maintain the health and the activity of the immune system. Now, the autoimmune disease occurs here in the pathological autoimmunity. So in the pathological autoimmunity, it's not just a training session. It is actually a real damaging immune reaction that goes out of control, leading to damage of tissues, producing signs and symptoms. So this is the autoimmune disease. And it is this category that we are going to deal with or talk about during the course, presenting different examples of autoimmune diseases. So immune diseases or autoimmune diseases are classified into two groups, either organ specific, or non-organ specific. In organ specific, the antigen presented by a T cell or the autoreactive lymphoid cells are directed against particular cells. So they attack these cells present in that organ. Examples including pernicious anemia, myasthenia gravis, type 1 diabetes, pemphigus vulgaris, vitiligo. So these are specific to one organ because these cells are targeted by the inflammatory reaction. In the non-organ specific, the self-reacting lymphoid cells are against certain structures or certain epitopes that are presented in different organs. This is why it is going to affect different organs and different parts of the body. This includes rheumatoid arthritis, for example, mixed connective tissue diseases, multiple sclerosis, Sjogren's syndrome, SLE. So these diseases are not restricted to one organ. It is actually affecting multiple organs, and the clinical picture of these diseases are different among patients. It depends on which organs are more involved and how severe the involvement is. So many of these conditions we will discuss in details in the coming lectures during this course. Autoimmune disease, the etiology of the autoimmune disease is not really a simple thing to deal with. It is considered one of the complex diseases. Complex means there's genetic factors, there's environmental factors, other factors that contribute to the etiology. For autoimmune disease, genetics are involved. The AIR gene, HLA, might be involved, and several associations have been reported. Environmental factors like stress, always stress is a it's not a cause, but it is a triggering factor. Many patients with different types of autoimmune diseases, their symptoms and signs will flare up during stressful situations. Age also plays an important role. Maybe as the patient gets older, you are more likely to develop one of the autoimmune diseases. The exact reason is not really known. Maybe with age, the amount of the let's say the control we have in tolerance is sort of become weaker. Number of T regulatory cells are reduced with age, so this is a failure of one of the tolerance control. Um, uh, you will have more time to allow self-antigens to get exposed to the self-reacting T cells, so you'll have more chance to develop autoimmune diseases. So age plays a big role. Usually autoimmune diseases affect middle and elderly age group. The gender also plays a role. Females are more likely to develop autoimmune diseases. Sometimes the ratio is 9-1 in some autoimmune diseases, like, for example, Sjogren's syndrome and or Sjogren's disease. The reason why there's difference in gender, hormones might play a role. Maybe there is other factors, trauma, infections. So we will talk about how these factors contribute to induce an autoimmune disease. Let's start talking about the genetics. So the R gene, which is a short for autoimmune regulatory genes. This gene codes for transcription factor, and transcription factor is the protein that binds with the DNA and start the process of transcription. So this transcription factor, the AIR, or the autoimmune regulatory transcription factor, binds with the DNA in the cells presented in the medullary thymic epithelial cells. Remember, the medullary thymic epithelial cells are those cells present in the medulla of the thymus and are responsible for presenting different types of self-antigens, including insulin, including all types of self-antigens. So this R gene, or this R transcription factor, binds with the DNA in that thymic epithelial cells and transcribing all of these genes present in these cells, giving products, proteins, self-proteins, variety of self-proteins that would be presented to the T cells during their training, during their negative selection. So when the AIR transcription factor is working properly, it will transcribe all of these genes, and the T cells would be exposed to so many self-antigens and would be deleted appropriately, and we will get rid of these self-reacting T cells during the negative selection. If this AIR transcription factor is not working properly, it will fail to transcribe these genes, so the medullary thymic epithelial cells would not be able to present self-antigens to T cells during the negative selection in the thymus, and in this case, many thymic T cells that have a self-reacting B cell receptor will get out to the circulation because they were not negatively selected. They were not deleted in the thymus, simply because they were not presented to these self-antigens. And those people that have a defective AIR gene and who fail to present these self-antigens, usually they have several autoimmune conditions or autoimmune signs and symptoms. They are all presented in this syndrome: autoimmune polyendocrinopathy, candidiasis, ectodermal dystrophy. So they have many endocrinopathy affecting the thyroid gland, the pancreatic beta cells, different types of endocrinopathies. They develop candidiasis and they develop different signs and symptoms, all related to autoimmune diseases. So this is one example of a single gene autoimmune disease. But this is not actually the standard, this is sort of the exception. And other autoimmune conditions, there are more of genetic contribution, but there is no one single gene identified as a cause of this autoimmune disease. Many associations have been linked to the HLA genes, which are the MHC, human MHC antigen presenting molecules. So for example, the type 1 diabetes has been associated with DQA1, certain polymorphism of the DQA1, and DQB1, O3, O2. So this is the type of the HLA. For example, multiple sclerosis has been linked to DRB1, 1501. Rheumatoid arthritis is again was associated with DRB1, 0404, celiac disease with another HLA type. So these are the strongest associations reported with HLA types. Why an HLA would be linked or would be associated to an autoimmune disease is the exact mechanism is not really known, but we know that these associations are real because they were reported in several populations. We have several confirmatory results supporting these findings. They were linked through family studies and associated through case control studies. So these associations are real and are not false positive associations. So we know that. But why would a person with a specific HLA type would have higher susceptibility to develop diabetes, for example, or multiple sclerosis? There are different explanations, different theories, but nothing proves these theories. One of them is maybe when you have a specific type of HLA, you have a specific antigen binding groove, and the shape of this antigen binding groove might, favor certain interactions with self-presenting T-cell receptors. So this is one of the explanations. Sometimes certain HLA types would have a specific shape or specific groove that favors binding to self-epitopes or self-antigens. So it will make this person more susceptible to develop autoimmune disease. This is another hypothesis. The third hypothesis, like some HLA types are not well presented in the thymus, so the T cell receptor that recognizes both HLA and the peptide would not have the chance to meet with this specific HLA protein complex or HLA peptide complex. So these cells would not be deleted and the autoreactive T cells would be present in the circulation. So these are hypotheses to explain why do we have these real associations with HLA or having the real associations of HLA with the autoimmune diseases. All of them are related to the three-dimensional shape and structure of the HLA binding antigen groove. The rest of the genetic studies are really sort of sporadic association studies. So you will have the certain group investigating NOD2 gene and find association with certain autoimmune disease, with diabetes type 1. this might not be replicated in the other group. Other groups will find association with another gene with an autoimmune disease. So the rest are sort of different association studies might not be confirmed, and this makes studying the autoimmune disease or the genetic basis of the autoimmune diseases is a bit complicated. Right, so this is about the genetic role in inducing autoimmune disease. Let's talk about infectious agents. So this is sort of interesting mechanism in inducing autoimmunity. And actually we are going to discuss lots of mechanisms suggested to explain the association of having particular infection with developing an autoimmune disease. These are the 5 suggested mechanisms for developing autoimmunity related to infections. The first is molecular mimicry, presence of inflammatory conditions, epitope spreading, release of cryptic epitopes, bystander activation. So we are going to explain each one separately. Starting first with the word epitope, we need to know what an epitope means so that we can understand the rest of the mechanisms. An epitope is a part of an antigen where that interacts with the antibody. It is very small parts. It's only 5 to 6 amino acids. So, for example, this is an antigen, and this is an epitope, which is a small part of an antigen, 5 to 6 amino acid, and this is the part that interact with the antibody. So an antibody would interact with not with the whole antigen, with a particular epitope. Usually one antigen would harbor many epitopes. So this is an antigen, and there are different epitopes, and each epitope can interact with a specific antibody that can recognize its 3-dimensional shape and structure of that epitope. So this is what we mean by the word epitope. Starting with the first mechanism by which an infection can induce autoimmune reaction is molecular mimicry. In molecular mimicry, this is a pathogen, and this is the antigen presented by a pathogen that interacted with antibodies. So we have antibodies that can react with this pathogen antigen here. And now these antibodies are circulating in the blood, and they can identify and they can recognize this pathogen antigen. If a self-cell has an antigen, a self-antigen, that looks very much similar, so it mimics the 3-dimensional shape of this pathogen. And remember that the antibody will bind with an epitope, so it just requires 5 to 6 amino acids to recognize and to make a binding. So if we have this 5 to 6 amino acid sequence, not necessarily to be identical with this epitope, just very much similar with this epitope, this would be adequate for the antibody to bind with self-antigen, because it looks like it mimics the pathogen. The best example of molecular mimicry is developing rheumatic fever after strep throat. So when a patient have Streptococcus pyogenes infection affecting the throat and upper respiratory tract, of course there's going to be an immune reaction. Lots of antibodies would be formed against the M protein, which is present in the strep pyogenes bacteria. So these antibodies will bind with the M protein of the pathogen. Now, there are certain proteins in the heart, particularly in the heart valve, that look very much similar to the M protein of the strep pathogens, bacteria. And even after the infection, all these antibodies are present, and they would be able to react with these self-antigens that are located in the heart valves and initiate an immune reaction there in the heart valve and in the sacral lemma of the heart. So the patient would develop rheumatic fever and would have problems in the heart valve, and also it affects other structures and other protein. So this is the best example, and it's not the only one. Other example includes type 1 diabetes that can develop after viral infections like hepatitis C infection, Coxsackie virus infection. So the antibodies produced against these epitopes, these viral epitopes, look similar to an enzyme produced in the by beta cells in the pancreas, and they would be destructed, inducing type 1 diabetes. Another example is the development of chronic arthritis after Borrelia burgdorferi infection that causes Lyme disease. So even when the Lyme disease is treated or after the infection, the patient still develops of chronic type of arthritis. This is not associated with the tick infection, with the bacterial infection induced by the tick. It is actually related to sort of autoimmune reaction because of mimicry or cross-reactivity. So these are examples of molecular mimicry inducing or causing an autoimmune disease. A second mechanism by which a pathogen can induce an autoimmune reaction is by creating an inflammatory condition. Remember, we talked about anergic cells, anergic autoreactive T and B cells. Now, these anergic cells, they do not create an immune reaction in the normal condition because these self-reacting receptors, T-cell receptor, can recognize the self-antigen, but this is not adequate to create an inflammatory response because it requires the second signal. However, when there is a severe inflammatory reaction, there's lots of cytokines, so many chemokines in the area where this anergic T cell is present. Now, these cytokines in the area surrounding this anergic T cell can activate it, would be adequate to activate it even without receiving the direct second signal from the self cell. So it would be activated and it would be able to interact with this self antigen and will create an immune reaction against this cell, which is a self cell, and this would initiate an inflammatory reaction. This is actually this explains that why many patients, after having, for example, a severe viral infection or severe bacterial infection, and they got hospitalized, they would tell you that after that period of severe infection, they had a reactive sort of arthritic pain, or they might have a flare-up in their arthritis, or they might have a flare-up of skin reactions or skin lesions. So these are related to activating anergic T cells, which promotes sort of autoimmunity. So this is the second mechanism of producing an autoimmune disease in relation to an infection. The third mechanism is epitope spreading. So what do we mean by epitope spreading? So let's see this image here, and let's follow these arrows and see what's going on. We have, this is the antigen presenting cell, and it is presenting a primary epitope or a pathogen epitope and presenting it to the T cell receptor here in this T cell. This T cell would get activated because it received this primary signal and secondary signal here. So now this T cell is primed and activated against this antigen. It will go from the thymus to the circulation, and from the circulation it will go here to the tissues. And now it should be able to interact with cells producing the same antigen, and will create an inflammatory reaction, releasing cytokines. These cytokines will recruit other inflammatory cells like monocytes, like macrophages. All of them will get activated, and we'll have an inflammatory battlefield here. And this inflammatory area would induce tissue destruction. In tissue destruction here, there's lots of tissue debris, there's lots of self-antigens that are released, and these self-antigens would be taken by the antigen-presenting cells and would be presented here in its MHC molecule. This antigen-presenting cell now goes back to the lymphoid organs, to a lymph node, or to the lymph node, and there in the lymph node it will present this antigen, which is a self-antigen released after tissue destruction here. So it will produce this self-antigen, and it will show it to the inflammatory T cell. Now this inflammatory T cell, of course, it has the ability to recognize this self-antigen, and it somehow escaped the central mechanism of tolerance. It escaped being deleted. So this T cell receptor, which was there dormant, ignored in the lymph node, now become activated because this antigen presenting cells has presented the self-antigen to it. Now it got activated, it goes through the circulation, it goes here to the tissues, and now it will react with self-presenting cells, inducing more damage, continuous damage to the self cells, creating an immune reaction. So this is what we mean by epitope spreading. Epitope spreading means after an inflammatory reaction, after damaging self cells due to severe inflammatory response, this would release different antigens. This would release different self-epitopes that become spread through the antigen-presenting cells, become spread to adjacent and surrounding lymph nodes, looking for T cells recognizing these self-antigens. One of the best examples of epitope spreading as a mechanism of inducing an autoimmune disease is multiple sclerosis. Actually, the presence of a viral epitope creating an infection, damaging the myelin sheath, might initiate an immune reaction against this myelin sheath by doing epitope spreading and creating this autoimmune disease. The fourth mechanism, which is release of cryptic epitope, actually nothing new here, but let's define the word cryptic epitope. Cryptic means hidden. So there is a hidden epitope that is not usually presented, not usually seen, but because of the infection, because of the damage, these epitopes become exposed and presented. This is an interesting thing. One example of an exposed or example of exposing cryptic epitope. This is an IgG, and when there is an inflammatory reaction, bacterial or viral infection, now this IgG will bind with this pathogenic epitope, and as it binds to this pathogenic epitope here, this will induce a conformational change, which means change in the shape of this immunoglobulin. And this change in the shape of the immunoglobulin would release a hidden area of carbohydrates. So these carbohydrates are usually like bent inside, so they are not seen, not recognized. They will be recognized only when this conformational change happens in this IgG. Now these exposed carbohydrates now are considered sort of a new epitope, a new foreign body that is recognized by the IgM. So this IgM is now recognizing these carbohydrates. These carbohydrates are creating an inflammatory reaction, and this is the initial inflammatory reaction. This is the IgM. Remember that it consists of five, or its pentamer consists of 5 units. And this IgM now recognizes the carbohydrates here, which is normal, self structure, and creating an inflammatory response against this. Interestingly, this IgM is the rheumatoid factor that we measure when we are looking for rheumatoid, or checking for rheumatoid arthritis, or any type of rheumatic or connective tissue disease. Okay, the fifth mechanism is a bystander activation. What do we mean by that? Let's study this image here. So this is a pathogen got into the tissues, and this created an inflammatory reaction. There's lots of cytokines, and these cytokines recruited dendritic cells. These dendritic cells are now activated, and there's a tissue destruction here. And in the tissue destruction, we will see lots of antigens. There's these pathogen antigens and these self-antigens that were released after tissue damage. As we said, these dendritic cells would take a sample of all the antigens present at the battlefield, the pathogen and the autoantigens, and it will migrate to the lymphoid tissues. In the lymphoid tissues, as we stated before, it will present both the pathogen and it will present the self-antigen. Here, if we have a T cell that has already escaped the central tolerance, they would be able to react with these self-antigens. So this is what we mean by bystander activation. So we mentioned bystander activation previously, and actually the whole 3 steps, the epitope spread, or the exposure of encryptic epitope here at the site of damage, and the epitope spread here to the local lymphoid tissue. And the third stage is the activation of the bystander T cells, which are the T cells in the anergic state that were ignored, neglected in the lymphoid tissue. Now they got activated by these dendritic cells. These activated T cells would go to the circulation and react to the self tissues and self antigens, inducing further destruction and damage to the self tissues. So going back to this image, we've mentioned the genetic basis of the autoimmune disease, discussed some of the environmental factors, explained the mechanisms, how infections can induce an autoimmune disease, medication, like they might act as an irritant to create an inflammatory reaction or inflammatory response. They might act as toxins that would aggravate or activate an inflammatory T cells. The last one is trauma or tissue damage. Actually, we have mentioned that some types of trauma that are related to infection, like tissue damage induced by an infection, would release some encryptic antigens. The same happens after trauma, like surgical intervention. If a patient had a surgery, the cut in these tissues might expose sequestered or hidden antigens, and if these antigens were located in, we call it immunologically privileged sites, these immunologically privileged sites are not actually accessible to the immune system or to immune cells, so usually they are hidden there in these privileged sites. But if a surgery took place, a trauma took place, and these sites, these antigens would be expressed would be presented to T cells, and this might create an immune reaction. Example of immunologically privileged sites includes cornea, anterior chamber of the eye, brain, uterine environment during pregnancy, and testicular tubules. So these are not actually exposed to the inflammatory cells. It's sort of protection of these structures from an inflammatory reaction. If for any reason they become exposed, this will induce severe damage in these structures. Actually, several cases of sterility in men might be related or induced by surgical procedures exposing testicular tubules and creating an inflammatory reaction that would damage the spermatozoa and sperms present in these testicular tubules. Autoimmune disease would take place when there are multiple failures at several checkpoints that took place. So there must be a failure in the central tolerance, and this needs to be followed by failure in the peripheral regulation. Other conditions including presence of auto or mimics to the autoantigens, or maybe damage in the tissues that induce presenting of encryptic antigens. And lastly, co-stimulation, either by abnormality or non-working Treg cells, or by presence of a huge amount of cytokines that is adequate to activate an allergic T cells. So an immune reaction would not occur because just of the presence of a self-reacting lymphatic cells. It would occur because of several, or because of cascade of failures, cascade of errors should take place for an autoimmune reaction to take place or to happen. Treatment of autoimmune diseases usually or generally started with steroid therapy. Steroids would suppress the whole immune system, which is actually not a good idea. However, we usually start with a steroid as the first line of treatment because of its quick action. But because of the side effects of steroids, the affecting the metabolism, affecting the bone, affecting the glucose level, so doctors usually tend to go to a steroid-sparing drug targeting specific parts of the immune system. We will talk later about certain biologics like anti-TNF or anti-interleukin 1 or anti-interleukin 6 medications that manage to target the certain part of the immune system instead of suppressing the whole immune system, which would make the patient susceptible to infections. We are trying to limit the side effects of these anti-inflammatory medications. So using biologics would help. Still, they have side effects, and our aim in the future, or the research actually try to figure out the mechanism by which this immune disease takes place. For example, is it related to epitope mimicry? So if it is the problem with epitope mimicry, so what is that epitope? Maybe they can create an antibody or monoclonal antibody that interact with that specific epitope and block it. In this case, we will keep the rest of the immune system working fine, working normally, and we will target that mimicking epitope. So this is the aim of the research, is trying to figure out the exact mechanism of the autoimmune reaction, the epitopes responsible for initiating that autoimmune reaction, and working or dealing with these specific epitopes to control the disease. The images presented in this lecture are taken from these references. This is our textbook. I love textbooks, so and I depend on them a lot. in preparing the lectures. So this is our main textbook. I have this textbook as well, and I have it in a PDF file, which I can share with you if you'd like to. And also, this is another reference from Natural Reviews Immunology, from Nature Reviews Immunology. And so I must state the references from which I took the images. And that's all regarding autoimmune diseases. And hope you enjoyed this lecture. Please let me know if you have any questions. Contact me, email me, and see you next week. Thank you very much. Bye-bye.